Good morning. Thank you to give me the opportunity to talk to you about our research on improving adaptive trait predictions on large geographical scales using genomic data. First of all, I would like to congratulate the organizers. It's not a small merit to put forward a conference in these times, and so I'm very happy that I'm part of this. So I will start giving a little bit of background about how to assess population responses to climate change. Of course, this is not a new topic. There have been a lot of work done, mostly based on the long history of provenance trials we have in forestries. And this work has been normally based on models that were using the information from common gardens to separate the genetic and plastic components of trait variation and project this into the future using different climatic models. And then that has been very successful. But now we are experiencing a new situation where there are very large genomic datasets that are being generated for several forestry species. And we have still to learn how to integrate all this information into the standard prediction models based on phenotypic traits only. So there are different ways of integrating genomic information in prediction. There are some that are pretty simple or relatively simple. Like for instance, this is a very interesting paper where basically they use genotype environment associations to identify different ecotypes of, in this case, well, it's not trees, it's bats, but it will work the same in trees, as we will say later. So they do this analysis just to identify different ecotypes, and then they use models that either consider the full species, the distribution of the full species, or they consider separately different models for each of the ecotypes. And we can see that the predictions are quite different. Actually, the predictions, when you consider that there are different ecotypes or gene pools, they are much more optimistic than the ones that are based in all species behaving in the same way. This is another interesting case where this is only based on genomic data, and it is also in trying to identify which populations are more vulnerable to climate change just by looking to how current allele frequencies in the different populations will match the allele frequencies that could be expected to be optimal in that location in the future. But for forestries, where we have very often in common gardens, we have even more relevant ways of integrating genomic information. And then when we can base our work in previous analysis of genotype phenotype association, what is called GWAS studies. And then we can look to model species like biodiversity to see how we can do. So basically these studies will start having association study, and that allows to identify the genomic variation that is underlying a trait, and then they will use that to map the future evolution of the trait. So this is a very landmark study that came out last year, and we have some first tries to do something in the same lines in forestries, like this study in Valley Oak, where they are using this GWAS information to compute some polygenic scores that could be used later to identify which plant materials will be more useful for plantation, considering adaptation to future climates. So given this background, our questions were first, how best can we integrate genome information into prediction models, in particular for those species that are of interest in our lab. And then if like having genome information provides an add value with respect to the current modeling approaches. And then for that, we need also to understand better, we want to understand better what are the underlying causes. That's it, the reasons why there is phenotypic variation in natural populations are large geographical scale, because that will allow also us to identify what are the best explanatory variables in our models. And all that, of course, will be used in the future. They will be useful to do predictions on how, what kind of genetic change, how much genetic change populations will need to do if they want to stay in the current locations and track the environmental changes that are taking place. So this is quite an ambitious research, but we were lucky to have a very good case study to develop. That is Maitland pine, Pinus pinaster. And this species is interesting because it has a substantially large distribution. It's not like a Scots pine or beech, but it's not restricted locally. We know that there is complex patterns of gene flow and local adaptation, but still it's a species that have a strong population genetic structure. And they have been, like you can see here in this figure, there are clearly different geographic groups that have different genetic composition. And that will help us to test different more factors that in other species. So, and for these species, we have a very large common garden that is based on clones in the clonal replicates. This common garden consists of 523 clones coming from the full different provenances of the species. And we have data for several phenotypic traits, but I will present here just the work done for modeling based on height measurements, over 30,000 of those. And this is the work done by Juliet Archambault, that is a PhD student supervised by Marta Benito Garzón and myself at INRA in Bordeaux. And of course, the phenotypic information is complemented in this case by different sets of SNP markers that have been genotyped on the same clones for which we had the phenotypes. There have been some modeling done in Maitland pine previous of a study. In particular, these two studies that I'm presenting here. In the first one in 2015 is something very similar to the work on the forest bat that I was showing before. And in this paper, what they are doing is to try to see what will be the future distribution of the species, either considering the full the data of all the gene pools together, the full species, or separating, doing different models and combining them for the different gene pools in Maitland pine. And you can see here the predicted distribution for the future could be larger. You will see in the yellow regions here, if you consider the differentiated gene pools, like in the case study for the bats. And then there have been also attempts to predict what will be the future evolution of quantitative traits, and this is for height, the same target trait we are working here. You can see that there is a positive evolution for population will grow higher, taller in height in the core distribution, but also there will be severe consequences for growth with less growth in the margins of the species. Still, this is the model that only includes, in this case, distribution data. In this other case, it includes phenotypic traits from common gardens, but it doesn't include genomic information. So we work with Juliet trying to find if by using genomic information, we can learn more and we can produce better predictions. This is not easy, so we started comparing models that include different factors, and at the end we finished it by comparing 12 models for high growth, and then but these models were each model were trying to answer a different question. I'm not going to present here the models. I will just show that we have some that is just baseline models, that is the standard genetic quantitative models that will allow us to separate genetic from plastic responses. Then there is a set of models that they are including climate information and also genomic information. And there are other set of models that they are just including genomic information to see if using only genomic data, we can still do good predictions. And then all these models were compared for its the predictionability using standard methods like I-square or the mean predicted errors. I will show what is new here from previous work is that we are using this genomic data. It's not only the trace measured in the common garden, and we're introducing this genomic data in two ways. One is as something that has been done before, that is just considering these distinct gene pools, like this, and this is the models that are here. But then we developed another kind of models that they are based in a previous GWAS study, and that they will consider counts of positive alleles. And I will explain what is that in the next slide. So this is what how we did to identify these positive alleles. So we have some marker data, and we developed some genotype phenotype association study, and we did it in two ways. We will see later how. So, but what we did, the core of we did, they have a lot of refinements, but in practically we just tried to identify these SNPs that were associated with height, and then identify which is the allele from these markers that will confer higher height to a clone or to a population. And then we counted the number of these beneficial alleles in each genotype in the common garden. And then we did it, as I say, two ways. One is using all the common gardens together, and that will provide some positive alleles that have a range-wide global effect on the phenotype. And then we use it also different GWAS studies for the different common gardens that are representing different environments. So this gives some environment-specific associations that allows us to identify alleles that are only important in a particular environment, but they will behave neutral in the rest of the distribution. And with that, we started comparing models, and I will start showing you what is the standard quantitative genetic analysis. That is our baseline analysis, and you can see here on the left, you can see the performance of the different populations. You can see that there is quite difference in growth between them, and we put some color. We are not using here the gene pool information in the models, but we put some colors here for the different gene pools for visualization. And you can see that there's a trend for populations from the Atlantic range to grow more, and populations from the Mediterranean range to grow less. But you can see also cases like these two populations in Corsica, where they belong to the same gene pool, they have exactly the same ancestry, but one is growing very tall, the other is growing very short. So, and this is because they are growing in very different environments. So, and you can tell that this one is pooling with the Atlantic population, this one with the Mediterraneans, and even when they are in the gene pool, in the same gene pool. So this is also giving us some evidence that there is some climate adaptation on top of whatever effects have to belong to a particular gene pool. And then on terms of plasticity, that's it, the difference between sites, we saw that there is a huge difference of performance between sites. And if you see to the scale here, compared with the scale here, you can say the plasticity actually, it has much more importance than the genetic component for this, for high growth. Well, that was the baseline model, and now we started to compare the other models against this. And I will present here only our best model that is interestingly, this model did not include either the provenance effect or the clone effect, but it could get a very good prediction just based on gene pools, climate, and genomics. So to start for the first factor, you can see here the effect of gene pool. It's very clear that the some gene pools have a very strong impact on growth. So just by belonging to a gene pool, that means that you will have a high probability of being taller than by belonging to the gene pool. But this is not the full study. We have also these other significant effects that are basically the effects of the positive effect alleles. And in this graph, you can see 5 distribution. one per each common garden, because also these effects were different depending on the common gardens. So depending on the environments where the effects are assessed. So you can see here that's the effect of genomics, and this is the effect of two climate variables that were also included in the model, precipitation and temperature. And I said what is very interesting is that the model includes only the climatic, the climatic data from the natural population, from the size where the natural population have been evolving. So they are is related to adaptation to the particular climate. And they use also this genomic information that is either the gene pool information, that is neutral information, is just information not related to adaptation, but just to the demographic history of the different of the species. And they include also these pairs, these positive effect alleles, that this is directly related to genomic markers that are associated with the phenotype. So, and for that was very interesting for us, but we wanted to know that that is the best model, but how much different is from the other models. So we came to this kind of visualization. So here you have a selection of models. In orange, you got the standard baseline models, that is just the standard quantitative model that takes into account only the provenances and the clones effects. Then you have a set of models that take into account the climate or the population and the genomics. And then you have a few models that take into account only the genomics, only the positive effect alleles. And what is quite interesting here, and I find it's quite amazing, is that if you see some of the common gardens, mostly the common gardens where trees are under very good growing conditions, actually most of the models are performing the same. So they have the same accuracy, the same predictability, and you can see that for Asturias and Portugal, and a bit for Bordeaux, a bit less, but still in Bordeaux. But then what is interesting is that when you see the common gardens that are under very harsh Mediterranean climate, you could see that the standard models are fully failing to predict the growth in these common gardens. And then those models that are including climate information, or they are including also like the genomic information, and are much better for prediction. And I did not say, but this is prediction of hair growth or new provenances. So that means that we took a partition of the data with a number of provenances to construct the models, and then they use this to predict the performance of the provenances that cannot be included in the model. And this is very good news, because it gives us a way to do predictions for populations that are not included in the common gardens. And to conclude, we'll say that what we have found, and there are some results that just validate things that we knew before, like we got again this very clear result that plastic responses are normally larger than genetic ones for hair growth. And what is a little bit new is that we found that there are these plastic responses, they are not only due to climate, but when we include sites in the models to replacing climate, they will have higher explanatory power, telling us that actually there is more than climate. Some populations and environmental characteristics in the sites that are not climate that was important. Then we saw that at least for maritime pine, where there is a strong population structure, the existence of distinct gene pools and the population demographic history is important. Different gene pools grow in different ways, and this is not related with adaptation, and perhaps partially, but there is, because there is a confounding effect with the potential effect of climate and the existence of gene pools. But it's also, it's not all because of adaptation, it's also because of the demographic history. And there is, again, a clear evidence of, on top of that, there is a clear evidence of adaptation to climate. And that's, I would say, that the best way to to see it is when we look to the Corsican provenances that come from the same gene pool but have different performance. And this is clearly associated with climate, and that's is something we cannot explain by ancestry alone. And there are other examples like the Leiria population in Portugal and many others. So what we have here is actually a combination of demographic history and adaptation to climate. And we know also that climate is not for history, but we should start studying other environmental variables, environmental factors that could be important, like soils or even the biotic environment. More conclusions is that our base model combines population demographic history with the provenance climate of origin, mostly including both precipitation and temperatures, and the height-associated positive effect alleles. And as I said, this is very interesting and a bit surprising because it allows us to do predictions actually without having data from common gardens. We found something that is also very interesting is that these positive effect alleles that show an environment-specific association, the regional pairs, they have more predictive power than those that are identified when using all the 5 common gardens together. So that means that we are looking to alleles that are neutral in most of the distribution, but they have just some importance in a particular environment, and that goes very well with the current theory that shows a predominance of conditionally neutral alleles. And just to finish the conclusion, just to say that it's interesting to see also that even models that only include genome information have quite good predictions, and that's again is very encouraging to work with a species for which there are not common gardens or there are a limited resources to establish them. And just to finish my presentation, I would like to give some perspectives of what is the future work we are doing in this line. We would like to develop this modeling framework for the 3 species, in particular species with different life history traits of different population genetic structure, because we don't know how much of what we get in my campaign is due to the strong population structure we have in the species. So it may be a very particular case. And so for instance, it would be very interesting to do this kind of studies in the Scots pine. And for that, we have there's a new European project that is called For Genius that could be starting in January, and where we will have the opportunity to test this model in Scots pine and beech. Also, we got some insights here from the new generation of experiments, and so we came to the conclusion that perhaps what we will need to do now is just to work with just a representation of populations from each gene pool, but then test more genotypes, and most importantly, to test them in many more environments. If we were to get through realistic spatial predictions, we will need to have a good representation of all the environment where these species will be expected to grow. So, and finally, we are establishing and I'm working with a very special case study that we believe will give us very valuable information. So we are focusing in a common garden that includes 30 populations from Corsica. And this is it's a very large experiment, but it's also very interesting because we have many populations from a single gene pool. So that is a case where population genetic structure is not relevant. And they are living in an island with a lot of environmental variation. So it will allow us first to construct models without population, where population structure is not important, and we could allow us to test and validate the models we have constructed and I have presented in this presentation. And this is all. So thank you so much for your attention, and I will be happy to have questions now. Thank you.